welcome back to the lecture series in animal cell culture or cell culture technologies. So, the first week we introduced the subject and then we went on exploring the different aspect of the biology of the cultured cells. So, if you recap, we when we talked about culturing the cells, we talked about take a part of the tissue, make a single suspension, and then you culture it on a dish. So, first thing which has to be taken into account, what is the demand of the cell of these cells for oxygen. So, we talked about some kind of a condition, creating a condition where proper oxygen to carbon dioxide balance is being maintained. Then the dish where you are growing the cell should mimic the extracellular matrix of the cell in vivo condition, that is inside the animal, and we talked about the role of extracellular matrix, how these cementing materials plays a critical role in determining the fate of the cell. Post that, of course, while talking about the adherent ring of the cell, we talked about classification. One has to understand, is it an adhering cell we are culturing or we are culturing a non-adhering cell. Okay. Then followed by this, we talked about the cell cycle progression. Are we dealing with cells with limited time division, or we are dealing with fully differentiated cell, and we introduced some of the terminologies: cell cycle leading to cell division, followed by differentiation, and at times de-differentiation, where the cell loses its as per the definition permanently loses its function which it is supposed to do post differentiation. Then we talked about de-adaptation, where the cell which is supposed to produce a specific function de-adapts or does not perform it under specific conditions, but that could be rectified. So, in other words, de-adaptation is a reversible phenomenon. So, these are some of the points, and finally, I insisted you to understand that how many passage a cell line, suppose you are using a cell line which has been immortalized, or it can grow in number of cycle, but then to a fundamental question come, what is a practical number really a cell can be used over possible division what we are utilizing. So, one really has to know that, because otherwise through every passage a cell goes through a life cycle and the cell loses some of its telomere and eventually lot of its function kind of gets compromised. But then it all depends how you want to treat the cell and for what purpose you are growing the cell. Are you using it for specific sensor applications, or you are using it for understanding certain event happening at the cellular level, or you are using it for some kind of hybridoma technique like antibody production, or you are using it for production of some other XYZ chemical. So, that particular aspect will determine how much careful you have to be with the cell type you are dealing with. Okay. So, let us resume the second week by kind of summarizing the aspect what we have already dealt and couple of other aspects which we have not talked about. Okay. To start off with, so we are into week week 2, lecture 1, W2, L1. So, talking about biology of the cultured cells. So, the point we have already dealt is oxygen and CO2 environment, whatever we are maintaining. We talked about extracellular matrix, and we talked about adhering and non-adhering cells. Then we talked about cell cycle progression. So, where we have mitosis phase, G1 phase, synthesis phase, G2 phase, and talked about cell division. Then we talked about differentiation, then differentiation and de-adaptation. Okay, this is what we have already covered. This part is there with us. There are a few more points in order to completely appreciate the biology of the cultured cell, what we'll be dealing with. And one of the very critical important thing what we'll be dealing with is the pH. Under what pH a cell is adapted to. So, generally we know our body pH is at 7, but while you are growing the cell, there are, or let us put it other way, why the cells are growing in your body, you have this blood which is flowing through, you have these extracellular matrix proteins, extracellular matrix solution, which is continuously washing out that spot and ensuring it to make a pH 7. So, when you talk about cell culture, just for your imagination sake, think of it. Here we have a dish, and in that dish we have these cells which are growing, this half moon shape, these are the cells, and here you have the medium in which they are growing. Now, how one can handle the pH of this medium, and how do we know what kind of pH these cells will prefer? Why I am telling you this? Say for example, we have to culture the cells of the stomach, which digest the food at a very acidic pH. Of course, from our knowledge of physiology, we know that this acid production by these cells, parietal cells, Schiff cells, which are present in the, just for those taking this example, in the stomach, which functions at a very acidic pH. So, it means less than 7. So, the cells which are involved in this is parietal cells, Schiff cells, all these different cells. These cells can withstand at a very low pH for a transient period of time. Because of course, in the stomach it is not that it is a place which is filled with acid. These cells specifically produces the acid by reacting the hydrogen ion and the chloride ion. Okay. But what is important for us to realize that it is not every cell, and they utilize this acid to break the food, and soon after that this acid is kind of again reabsorbed or kind of it was destroyed. So, for a transient period of time, if I have a pH scale out in the stomach, if you think of it something like a pH scale here, where, so if I say these are, this is 7 and this is plus 7 onward, these are my below 7. Okay. So, for example, less than 7. So, what will you observe is, and this is the of course the acidic side. So, from the baseline value during digestion, this environment milieu goes like this for a finite period of time. This thing happens. So, then what you interpret from this is the cells in the stomach can withstand a very low pH for a transient period of time, but in order to regain that kind of activity, we have to create such a situation. So, if we are culturing these kind of cells, we should know that what is the level they can withstand. Similarly, there are cells, if you bring them down to that kind of pH level, they will die. They will not be able to withstand that kind of high proton concentration, right. So, maintaining the pH of a system is extremely critical, and knowing the window to which a cell can withstand it, because when we are growing things, we are not removing all other debris which the cells are producing. That kinds of create a situation where the pH may automatically fail. There is always a possibility, and there is always a danger in the cell culture that your culture dish goes on an acidic frame. So, this is something which one has to be very careful. This is one aspect. Second aspect in that line is that we have talked about the pH now. To add up what all we have talked about, so here we have now talked about the pH. The next what we will be talking about is, say for example, here the cells are growing in a dish, adhering cell, assuming that these are all adhering cells, and these adhering cells are dividing. Okay. And this is the medium where they are growing. Now, under this condition, these cells which are dividing will need energy, and its only source of energy is the medium, because none of these cells are synthesizing their own food material. They are depending on the medium to supply them with energy. Now, two things we are introducing. One has to know what kind of medium we are going to use, because though the basic salt solution remain more or less same, but at different spots of the body, different parts of the body, there are several other added molecules for specific cell types. these are cell type specific. You might wonder how that is happening. So, such thing happens, say for example, if you consider your body, just that old analogy what I gave you, same, for example, these are colonies of cells in your body, some are red, some are green, okay, some are blue, like this, and blood vessel is traveling like this, okay, which is bringing the necessary nutrients out here. But still, every cell type has a specific requirements. So many of the factors which, say for example, these colony of blue cells will be needing, their surrounding brothers and sisters secrete those, and through blood vessels or through direct contact, these are being transmitted to these cells. Or blood brings specific, say, hormones or growth factor whose receptors are very unique on specific cell type. So in a way, again coming back to that fundamental concept that cells live in a very dynamic environment, extremely dynamic, extremely changing. Whereas whenever we do a culture, we essentially follow a very static model. At least as of now, the most of the models which are being followed across the world are very static models. So each cell type possibly needs, if we are growing a pure culture of a specific cell type, say for example, I grow these blue cell as separately, or the red cell as separately, or the green cell as separately, I may need to provide those specific molecules which are cell type specific. They mostly come in the form of hormones and growth factors. The complexity arises if I have to grow, say for example, two different cell types together. Under that situation, say green needs a different kind of growth factor and red needs a different kind of growth factor. So how I ensure that the growth factor received by green, which I am providing from outside out here, does not interact with the red? There is only one possibility. Either red does not have a receptor for it, then you are very lucky, or red will have a receptor for it, and that adds up to the complexity. In a real life, what happens? There are blood vessels which are crawling through, and they ensure that these binds there. But outside the system, that privilege is lost. So those adds up to the complex situation of growing cell in a very, very controlled environment. Okay. Further, we talked about the energy molecule. So every cell, if you look at it, mostly their readily sourced energy source is glucose. So every medium what we develop will have glucose as one of the major source of energy, and moreover carbon. But that comes with some set of problems which will be coming later. We use glutamine and other sources. There are reason and rhyme. So with this, I will close in onto this part. I will add that last tail piece, what all we have covered. So we talked about oxygen and carbon dioxide. We talked about extracellular matrix. We talked about the cell cycle, de-differentiation, de-adaptation, and of course we talked about the cell lines, and of course in between we have talked about little bit, very little bit about primary culture. Okay. And now we talked about, then we talked about the pH acidic or basic pH, whereas most of the cells grows at 7, pH 7. And then we talked about medium, growth factor, hormones, and the need for energy molecules or source of carbons, or source of carbon. So, these are the very basic fundamental what one has to keep in mind while one is planning to do cell culture. And if you have the basic knowledge of physiology, then these things should come very handy. Okay, fine, what all my body needs, that is the same condition or close to the same condition I will have to provide. Now, if you remember, when we talked in one of the very early classes that whenever I am growing the cells, so say for example, this is my knowledge of in vivo physiology or physiology which is happening inside the cell, and here I am with in vitro physiology. What we talked about is in vitro physiology in the form of oxygen CO2 tension, extracellular matrix, pH, cell cycle, medium, culture medium of course, and in this you have energy source, growth factors, and hormones. Okay. Now, what is critical for you to know is after growing the cells in all these conditions, which is our goal is to come as close as possible, almost similar, almost congruent. Then what we have to do, the cells which are growing here, their properties have to be evaluated. Say for example, this cell produce certain level of activity of an enzyme. Say for example, it has an antioxidative enzyme capacity and it produces a P unit of it. Now, I have to compare it with in vivo condition, how this cell is functioning and what is the quantification parameter of this particular one property or many properties as a matter of fact. And if under in vivo condition, say for example, if I say activity in the y axis, so if the activity of a particular thing is somewhere here, say for example, how far I am in vitro condition, am I exceeding it, am I below it, am I same at it. So, depending on where am I have to understand the biology of this cell which is undergoing in an in vitro setup, or I am not even performing that function, I am here 0. So, this site, the one I am circling with red are the one which are the in vitro physiology of the one imaginary aspect, the P unit, what I am trying to tell you. Okay. So, there comes the real challenge, how much you have understood the biology of the cell, of the cultured cell, and how much you have appreciated the in vivo physiology of that particular tissue from where you have derived the cell sample. In order to create an artificial condition, artificial or synthetic growing condition. Fine. So, understanding of the basic physiology and correlating it with in vitro physiology is very important for a successful execution of an experiment, validity of the data in real life, and understanding the shortfalls or shortcomings what you are undergoing while performing this task. So, with this is our first class in the second week. We will move on to the layout and design. So, next our objective will be to move on to, with this basic start off, I will move on to layout and design of a cell culture facility. Okay. So, I will close in here. Please go through the points and think over it, ponder upon it, and develop your own philosophy about the subject. Okay. Thank you. Thanks for your patience listening.